What was the most exciting moment, would you say, in your research careers? I mean, you both sort of made sort of a number of contributions, but what was the thing that really turned you on?
Well, for me, there were sort of two moments in my career when I just was unbelievably excited by what I saw in the microscope. And, you know, as one moves up the academic ladder and, you know, has a lab of one's own, you don't have those moments anymore because you're busy supervising other people and listening to what they've done, and they get those moments. But for me, there were two moments when I just looked in the microscope and thought, oh my God. And one of them was when I was working as a postdoc, trying to grow these embryonal carcinoma cells from the tumors. That was sort of the first step, was to try to take the tumor cells, put them in culture, and make them proliferate and see what they could do. And I remember it was at University College London. It was very cold. I had chilblains because they turned the heat off. And
it was to keep the research costs low, of course.
And I was looking in the microscope, and I noticed that the cells that I had been culturing had clumped up and had floated off the dish and had started to differentiate. And it suddenly struck me that what they were doing was trying to be embryos. And these were these tumor cells that, you know, if you keep them flat, they just proliferate. But if you allow them to clump up, they just start to make embryos. And the sudden realization that these cells were trying to be embryos was really, really exciting. And then the other moment was when I had tried to put normal embryos in the dish and saw that they were growing just like tumor stem cells. And I don't think there's any substitute in one's scientific career for looking in the scope or whatever your particular, you know, seeing a gel or whatever, and suddenly understanding what's going on. And that doesn't happen once you move away from the bench, I think. So it was very rewarding.
Liz?
Well, probably quite a similar story in a sense, because when I was a postdoc working together with Alan Bradley, we used to do all the we used to look after our own vermin, let's put it that way. And so every Monday we would work in the animal facility and look at all our cages of animals and score all the offspring, all the progeny. And we had a very simple experiment. We were taking these wonderful chimeric mice that were a mixture of the embryonic stem cells and a host embryo, and so they were black and white. And what we were looking for was the observation that these stem cells, which were made from a black mouse, would actually contribute to the germ cells, and therefore these animals would produce black offspring. And so every Monday morning we would look through our breeding boxes and we would say, I wonder what's going to happen this week. And there was one very famous Monday morning when indeed we took the lid off one of these cages of mice, and there was a litter of these perfectly beautiful baby black mice. And we just thought to ourselves, gosh, you really can get this to work. And it was one of those, you know, great moments that I think both Alan and I will remember for a very long time. Kind of proof of principle, yes, it will work. And then a second kind of similar story in a sense was many years later when we began to understand the principles of how you take these very simple embryos and establish pattern, and head and tail of the embryo, the anterior and the posterior pattern, was we'd made a mutation in a particular molecule that was responsible for transducing a signal from a growth factor. And this is actually an experiment I did with my colleague Liz Bykoff, and we started to look at these embryos and dissect them, and we had a hypothesis that the extraembryonic tissues of the embryo would be very important. They're not just throwaways. And all of a sudden, we realized when you start looking at the phenotypes of these embryos and how they were disturbed, that yes, indeed, this model was perfectly correct. And so again, it's always nice to have a model and then to do an experiment and prove that your thinking was right. So again, just like Gail was saying, it's looking at the experiment itself and looking down the microscope, and it's a real thrill to have that real buzz. It's a real buzz.
I've got another question. This is for Cori as well, actually, all three of you. I mean, where do you see this field going now? I mean, you've made these contributions which really have allowed us to do quite extraordinary things. So what's your, looking in the crystal ball, what do you think is going to happen over the next decade or two? Who wants to have a go at that one?
I think Cori should get
No, I think the mouse is really developing into the workhorse of biology and biomedicine. And as we start to understand aspects of disease in humans, moving right away to the mouse to start to be able to just tear them apart is something that's an active area of research. And this has been the case for cancer, for diseases that we're starting to study. It's starting to be the case now for neurological diseases, some of the diseases of aging, and people are hoping, in the future that this will also be the way that we can understand psychiatric diseases. It sounds pretty crazy to study psychiatric disease in a mouse, but with the genes, which are like a Rosetta Stone, where you can really understand that the gene in the mouse and the gene in the human are doing similar things, the mouse becomes incredibly powerful as a way of guiding us along our path. So there's a medical importance of this work, but all of that medical knowledge really does not exist in a vacuum. There are times that we can do something medical without understanding biology, but those times are rare, and increasingly we really need to understand what the biology is in order to develop the medicine. And the biology that we've learned about what a mammal is from studying these kinds of artificial mammals, in a sense, that nonetheless have the same rules as underlie all of our development, is the knowledge that it's the system that just keeps giving scientifically.
You're in New York, nothing's crazy about psychiatry in New York, even psychiatry with mice, I'd say. Yeah. Who else would like to have a go at the future?
Well, I guess when embryonic stem cells were first isolated and embryonal carcinoma cells before them in the 70s and 80s, it was really exciting to have pluripotent or totipotent cells that you could watch do things. But in those days, it was very difficult. People tried many, many times in many ways to direct the differentiation of those cells into specific things. And really, those studies were remarkably unsuccessful. And I think, of course, the reason was that we didn't have markers. We didn't, maybe we were getting these special cell types out of these cultures, but there was really no way to identify them. And I think what is remarkable to me is reading about the efforts that people are making to direct the differentiation of embryonic stem cells into specific cell types that may actually be useful for approaches to, replacing organs in mice and figuring out ways to do that. Now it's actually starting to be successful. Both because we have molecules and approaches to use that weren't available then, and we have markers to know what we're getting. And I think that I'm just constantly surprised, having grown up in an environment where we couldn't do this, to see that people are successful at this now. And I think, I used to think it was 20 or 30 years off, and now I think it's not that far off, that cells like this will be useful for possibly replacing organs.
Liz?
Well, I'm going to come back to the point that Cori was making. I think it's a very useful possible translational tool. I still think the ability, now that we know the kind of linear genome sequence of many organisms, ourselves included, I think that, from my perspective, this technology has been and will continue to be absolutely fundamentally important for not only understanding what a gene does and how it works, but understanding the kind of complexity of our genomes and what regulates it, and really kind of teasing apart the ability to actually go in. We can now go into the mouse genome. We can change a single nucleotide base pair in the genome. and look at the consequences of that. So when you look at the complexity of the human population and multigenic diseases and all sorts of very interesting genetic phenomena that we're confronted with every day, I think it's going to still be extremely useful to be able to test our hypotheses in the mouse and learn a lot more about us and our physiology from being able to do those kinds of manipulations. So I still think there's a lot we can still do with a mouse model that's going to be very relevant.
I'd like to end with one perhaps more general question. This is a prize aimed at women. So I think the audience would probably be interested in your views of what the position of women in science is today and how it has or has not changed over the years that you've been involved. And again, I think it's open to everybody.
Well, we were having a very, very nice discussion earlier this afternoon with the FACES organisation, which is I guess a group of women postdoctoral students and predoctoral students and MD PhD students here at the university and other associated universities, and we were exactly having this discussion. And one of the points that I was making was that I think we've been very fortunate, both Gail and myself, to work in an area where there has been historically a very large number of very prominent women geneticists and developmental biologists. And certainly, I think it's a bit like genetics. You know, you look to the former generation for guidance and for mentorship, and I think that working in an area where there's quite clearly been a lot of very good women leaders has been very helpful in that regard. And so when you compare it to something like mechanical engineering or physics or maths, I think that we've been quite privileged, and it's been probably easier for ourselves as women biomedical scientists than it has been in other disciplines, but that's just my perspective.
I mean, I think that that things have changed, certainly in the time when I was a postdoc and was not permitted to work in a particular institution because of a nepotism rule, which meant an anti-female rule, basically, to a time when women are sought after and accepted as equals in most institutions, maybe not all. And so I think that there's been enormous progress in the last 25 or so years, and hopefully will continue.
You're optimistic for the future?
Yes.
Yeah.
Good.
Cori, anything you want to add to that?
So I think, you know, if we had gone through, especially with Beatrice Mintz, we would have been sort of following a generation from someone where it was extremely difficult to be a scientist to an intermediate point that Gail is referring to, where there were still real institutional barriers to hiring, to going to school. It's, you know, it's sort of amazing what's happened even in our lifetimes. Yale didn't admit women until 1969 into their undergraduate school. It's astonishing. And so I would consider myself sort of the first generation where the overt discrimination is gone. And now I think what's really necessary is encouragement. And I think the encouragement of example, of looking at someone and thinking, maybe I could be like that, but also the encouragement that occurs that we all do as teachers, which is a component of what everyone does as a scientist, and as examples, I think are the place to keep building.
Well, I think that's a good place to stop. I'd like to thank our panel. I mean, great discussion. Thank you for that.